Welcome to the webinar today entitled Engineering iPSC-derived Natural Killer Cells for Enhanced Efficacy of Cancer Therapies. My name is Damani Bryant, a content developer at R&D Systems, a Bio-Techne brand, and I'd like to thank everyone for attending today. Our featured speaker is Dr. Bruce Walcheck, Professor of Immunology at the University of Minnesota. Before moving into the presentations, I would like to inform the audience that this webinar is sponsored by Bio-Techne. Bio-Techne brings together the prestigious life science research brands of R&D Systems, Novus Biologicals, Tokris Bioscience, Protein Simple, and Advanced Cell Diagnostics. Together, we provide the scientific research community with a comprehensive and world-class product portfolio of reagents, assays, instruments, and custom manufacturing and testing services. Our first presentation features Scott Schachtel, a product manager at Bio-Techne focused on stem cell and cell culture solutions.
Great. Thanks, Damani. Like he mentioned, my name is Scott Schachtel, and I'm a product manager at Bio-Techne focusing on stem cell and cell culture tools. And I just wanted to take a minute or two up front here to start and introduce you to some of the tools that Bio-Techne has for NK cell research. So natural killer cell culturing and characterization is really a strong suit for Bio-Techne. And as you can see from the workflow graphic on the screen here, we really have a good set of tools across the full workflow, from isolating and expanding NK cells through characterization and analyzing them. In the next few slides, I'll briefly expand on each of these workflow steps, highlighting some of the tools that we provide. For isolating NK cells, we have our MagSelect isolation kits, and these are available for both human and mouse. These kits use negative selection to increase the purity of NK cells in your peripheral blood mononuclear cell preparations. So as shown in the data example on the screen here, the flow plot on the right shows a clear enrichment of CD3 negative, CD56 positive cells following isolation using the MagSelect kit. As you move into the expansion phase of your workflow, R&D Systems, a Bio-Techne brand, has a full portfolio of high-quality cytokines for NK cells. Key cytokines such as IL-2, IL-12, and IL-15 are also available as GMP grade, which can help ease the transition between basic research and cell therapy manufacturing. And really one benefit of the Bio-Techne portfolio and all the brands that it contains is that we manufacture almost all of our recombinant proteins, antibodies, and small molecules in-house. And we've taken full advantage of that to produce all-in-one kits for immune cell differentiation, including natural killer cell expansion. So the Cell Vivo NK cell expansion kit in particular includes optimized combinations of cytokines and media, along with the detailed protocol for the feeder-free expansion of NK cells. And actually, Dr. Walcheck was kind enough to run our Cell Vivo kit alongside the K562 expansion method and found similar growth rates, and that's shown in the histogram in the bottom right-hand corner of the screen. And more information on these can be found at the URL at the bottom of the screen as well. So verifying marker expression is obviously another key step in the immunology workflow, including when working with NK cells. So between R&D Systems and Novus Biologicals, Bio-Techne has one of the most extensive portfolios of antibodies for NK cells. And a nice way to search for these is using our interactive pathways, one of which is displayed on the screen here. This one is for NK cell receptor interactions. And these pathways can be accessed through the URL displayed at the bottom of the page. So I highly encourage you to visit the website and browse through those. It's a nice way to get information as well as direct links to products, including antibodies, for your research. And finally, as you interrogate and experiment with your NK cells, Bio-Techne has a wide range of immunoassays and cell death assays that are available, and all these can be found at rndsystems.com/nkcells. And so hopefully that just gave you a nice broad overview of kind of Bio-Techne and some of the tools they have in this space that can help your research and development. And just a few quick other things before passing the mic over to Dr. Walcheck. A couple housekeeping items here. We invite you to ask any questions for our presenter through the webinar or throughout the webinar using the Ask a Question box that's just below the presentation screen. And these questions will be asked to the presenter during the panel discussion or the Q&A directly following the presentation. In addition, in the upper right-hand corner of the screen, you'll see a tab called Resources. And in that tab, you'll find some extra content that relates to today's webinar. Okay, and so now I'll introduce our speaker for today, Dr. Bruce Walcheck. Dr. Walcheck studied at Montana State University at Bozeman, receiving his PhD in immunology in 1994. He's currently a professor at the Department of Veterinary and Biomedical Sciences at the University of Minnesota, and has faculty appointments also with the Minnesota Stem Cell Institute and the Microbiology, Immunology, and Cancer Biology graduate program. We're very excited to have Dr. Walcheck here today. And with that, I will pass the control over to him. So thanks for joining us today, Dr. Walcheck, and looking forward to hearing your presentation.
Okay, thanks, Scott and Damani, and want to thank Bio-Techne as well for this opportunity to discuss some of our work using induced pluripotent stem cells to derive engineered NK cells with enhanced function with the goal of deriving new and improved cancer therapies. So I was just going to start with a general overview of natural killer cells as I understand them. Our lab over the years has primarily focused on neutrophils, and over the past couple of years we've developed more of an interest with natural killer cells and trying to manipulate these cells in terms of killing cancer cells better. So the immune system is really divided into two arms. We have the innate immune system and the adaptive immune system. And we have different leukocyte populations in each arm of the immune system. For the innate immune system, we have neutrophils and eosinophils, basophils, monocytes, macrophage, and natural killer cells as the more predominant leukocyte populations. In the adaptive arm of the immune system, we have T cells and B cells. And so a real key distinction between the leukocytes within the innate and adaptive immune system is the type of receptors that they express to recognize pathogens or cells in the body that have become damaged, for instance, stressed, infected, or undergone transformation into a neoplastic cell. So each leukocyte of the innate immune system has a very heterogeneous specificity. That is, they recognize a broad array of determinants specific to pathogens or damaged cells. And each of the cells in the adaptive immune system has a singular specificity for the most part. So in simplified terms, really each T or B cell has a different antigen specificity, and the population as a whole has a very heterogeneous or broad specificity. So receptors expressed by the innate immune cells are encoded by intact genes inherited through the germline, whereas T and B cells express receptors that are encoded by rearranged gene segments, and this creates incredible diversity in their specificity. So NK cells were identified in the mid-70s, and it's now very well appreciated that they use an array of receptors to interrogate cells of the body as either self or altered self. And so in this slide on the bottom right, I show a density dot plot that indicates the staining of CD56 and CD3. These are hallmark markers of NK cells. NK cells are CD56 positive, but they are CD3 negative, which is a T cell marker. And in the peripheral blood, there are really two major populations of NK cells. They're CD56 bright or CD56 dim. And so in that density dot plot, you can see the majority, really greater than 90% of these cells are CD56-dim, and then a small population on top of them are the CD56 bright cells. CD56-dim cells are considered to be the mature NK cells, and CD56 bright are more of an immature population. And as a whole, though, NK cells in the blood are a pretty minor population. Neutrophils are the most predominant leukocyte population in the blood. They're about 60% of your leukocytes, where natural killer cells are about 2% of the peripheral blood leukocytes. But NKs are really emerging as an important cell therapy for cancer, which is indicated in the next slide, because they have this potent anti-tumor activity without a need for prior antigen sensitization. They're not MH restricted in the antigens they recognize. And upon adoptive transfer into the patient, they present a reduced risk for graft versus host disease. So even though NK cells are part of the innate immune system, they do have a lot of similarities, a lot of overlap with T cells in the adaptive immune system. For instance, NK cells are part of the lymphoid lineage, whereas most of the leukocyte populations that are innate immune cells are myeloid cells. NK cells undergo a tolerance or licensing, and this is similar to sort of the positive and negative selection process that T cells undergo in the thymus. And for NK cells, this licensing process is regulated by inhibitory or activating killer immunoglobulin-like receptors, referred to as KIR receptors. And so these KIR receptors are differentially expressed by NK cells, so it's a very polyclonal population. And you can get clonal expansion of some of these populations, as occurs with T cells. And even though in general NK cells are pretty short-lived cells, certain populations have demonstrated more of a long-lived capability following cytokine stimulation or certain types of antigen stimulation. So as I mentioned earlier, NKs express a very heterogeneous array of receptors, and these receptors interrogate cells within the body really in two ways. They're assessing determinants expressed by cells that signify whether the cell is healthy or altered in some manner, again, as it undergone trauma, stress, infection, or transformation. And so receptors on NK cells discriminate ligands on target cells, again, that indicate whether they're healthy. And by this approach, they're recognizing certain class 1 molecules on the target cells. These class 1 molecules, based on their expression, indicates that this is a healthy, normal cell. And then the NK cell utilizes other receptors that recognize ligands on target cells that can be upregulated when the target cell is stressed. And this is an indication of altered self. And so these receptors expressed by NK cells can transmit either inhibitory or activating signals into the NK cell. And it's really the balance of these inhibitory and activating signals that determine whether the NK cell gets stimulated or not. So if the target cell is in a healthy state, the ligands being recognized, such as class 1 molecules, are expressed at normal levels. The NK cell predominantly receives inhibitory signals and does not become activated. If the target cell is stressed in some manner and the activating receptors are being stimulated, then the balance of these signals tips towards activation and the NK cell becomes stimulated. And it performs really its two major effector activities. That's target cell killing, mediated by these factors that NK cells release, such as peripherin and granzymes. And they're also very good at cranking out various types of cytokines, in particular interferon gamma. And so interferon gamma can stimulate cells of the innate immune system, such as macrophage, as well as stimulate T cells. So NK cells serve as an important bridge connecting the innate immune system to the adaptive immune system. So then in this next slide, I tried to depict some of the scenarios that result in NK cell inhibition or activation, trying to make it more straightforward. I think the slide probably just came out to be a little busy. But what I'm showing here is on the left-hand side, again, NK cells are expressing inhibitory receptors. These can be these killer immunoglobulin-like receptors or KIR receptors, or C-type lectin receptors such as NKG2A. They're assessing class 1 molecules on target cells, and if the target cell expresses these certain class 1 molecules, the inhibitory signals are the predominant signal, so the cell tips towards inhibition, kind of indicated at the bottom by the signaling levels. It's primarily receiving inhibitory signals, and the NK cell is not activated. In the middle scenario, the target cells may have downregulated these class 1 molecules due to transformation or infection. This results in less signaling by the inhibitory signals. This is considered a missing self scenario. The inhibitory signals are not signaling at the same level, and now the balance tips towards activation, and the NK cell becomes stimulated. And on the right-hand side, I'm showing where the target cells have upregulated these activating ligands, and they're being recognized by activating receptors on the NK cell, such as these natural cytotoxicity receptors like NKP30, P44, P46. We also have NKG2D and CD16A, all very potent activating receptors. If they're getting engaged, the balance in the signaling, again, is tipping towards activation, and the NK cell becomes activated. In this next slide, I'm providing an overview of really the two means by which NK cells recognize target cells. There's this direct recognition, where the NK cell is using its activating receptors to directly recognize ligands on the target cell. This results in NK cell activation, and you get natural cytotoxicity and killing of the target cells. And the other example is an indirect recognition, and this results in antibody-dependent cell-mediated cytotoxicity. So here, an antibody is binding to the target cell. The NK cell expresses receptors that recognize that antibody. This activates the NK cell. It releases its cytolytic factors and cytokines, and this results in target cell killing. So this direct recognition or indirect recognition are really mediated by separate receptors. And our focus, our interest has been on antibody-dependent cell-mediated cytotoxicity. And on the left-hand side of the slide is an illustration where the primary receptor expressed by an NK cell that recognizes antibodies on the target cell. This receptor is CD16A. When it engages that antibody, when that antibody is attached to the target cell, this activates the NK cell, and again, it releases its cytolytic products and produces lots of cytokines. The bottom left-hand part of the slide, I'm taking you through the parts of the slide because I don't have a pointer that I can use, so hopefully you're able to follow me through these slides. And on the bottom part is shown the CD16A receptor. It's also referred to as Fc gamma receptor 3A. The R stands for receptor because CD16A is binding to the Fc region of an IgG antibody, so gamma represents IgG antibody binding. And on the right-hand side of the slide is the IgG Fc receptor family. There's really three groups or three classes of IgG Fc receptors. There's Fc gamma receptor 1. This is also referred to as CD64. It's really the highest affinity Fc receptor in the family. Then there's Fc receptor 2. There's three members in that family. R2A and R2C are both activating receptors, and R2B is an inhibitory receptor. And then there's Fc receptor 3 group, and that consists of an A and B members. CD16A, again, is expressed by NK cells. Really is their exclusive receptor used to recognize antibody and mediate ADCC. And CD16B is primarily expressed by neutrophils. So in that figure I show these allelic variants. There's two allelic variants of CD16A. And so at position, amino acid position 158, you can have a valine or a phenylalanine. And these result in different affinities in the receptor. CD16A is basically a low affinity receptor, but the valine variant is a higher affinity version than the phenylalanine version. And so this has clinical significance in that therapeutic antibodies that induce ADCC show greater efficacy in patients that are homozygous for the V variant of CD16A. And so therapeutic monoclonal antibodies have become one of the fastest growing classes of drugs, and tumor-targeting monoclonal antibodies are one of the most widely used and characterized immunotherapies for hematologic and solid tumors. And several clinically successful antibodies utilize ADCC as a mechanism of action. And so in this slide, I show three examples of some very well-known therapeutic antibodies that are widely used in the clinic for cancer immunotherapies. We have cetuximab, also known as Erbitux, the tumor-associated antigen that it recognizes is epidermal growth factor receptor 1. And this antibody is used in the clinic for certain cancers like colorectal and head and neck cancer. Then there's rituximab or Rituxan, which recognizes CD20, and this antibody is used in the clinic for certain types of lymphoma. And we have trastuzumab, also referred to as Herceptin. This recognizes EGFR2 or HER2 and is used in certain cases for breast cancer. And so these therapeutic antibodies can be used as targeting agents to direct NK cells to specific tumor antigens and malignancies. So an interesting feature about CD16A is it undergoes this regulatory process that results in its very rapid downregulation from the cell surface. So depicted on the right-hand part of this slide is CD16 sitting in the transmembrane of a cell. And in this cell membrane, there's a protease called ADAM17. It's a membrane-associated protease. It's normally in a resting state, but when the NK cell gets activated, this results in the induction of ADAM17, and this results in the clipping or shedding of CD16A, and this is referred to as ectodomain shedding, because essentially this shedding event occurs very proximal to the cell membrane and results in the release of the entire ectodomain of CD16A. And so the shedding process occurs, again, when the NK cell gets activated. This activation process can occur through CD16A itself, which again is a very potent activating receptor, or various other stimuli, other activating receptors expressed by the NK cell, or various cytokines that stimulate the NK cells, can all activate ADAM17, induce this downregulation process. So on the left-hand side, I just depict CD16, which is normally expressed at very high levels on a resting NK cell, following cell activation in various manners. In a very rapid process, CD16A is shed from the cell surface, and this results in a very rapid downregulation in its cell surface density. So CD16A signaling seems to be regulated in a constitutive manner by various inhibitory receptors that the NK cell expresses, but can also be regulated in a very rapid and efficient manner upon the induction of ADAM17. So it appears to play an important inhibitory checkpoint role in controlling the cell surface density of CD16A. And so ADAM17, just very briefly, is a member of this large family referred to as A disintegrin and metalloprotease family. There's approximately 30 members to this family in humans and mice. And again, this is a membrane-associated protease, and it has various functional domains in the ectodomain part of the protease. It has a metalloprotease or a catalytic domain, a disintegrin domain, and a cysteine-rich domain. And ADAM10 is probably the most similar to ADAM17 in terms of its structure and overlap in some substrates that it recognizes. But in terms of cleaving CD16A, ADAM17 really has a non-redundant role in that. It's the primary protease that mediates that event. There are various other members in this family that have a catalytic activity as well. There's various members that don't have a catalytic activity and more function as an adhesion molecule. So in addition to CD16 downregulation in vitro following overt NK cell activation, this process has been reported to occur in the tumor environment of cancer patients during the ex vivo expansion of NK cells for their adoptive transfer in the patients and in patients receiving certain therapeutic monoclonal antibodies. And these are all described in these citations that I provide on the left-hand side. So it's our assumption that CD16A shedding under these circumstances will decrease the efficacy of tumor-targeting antibody therapies. And so it's been our lab's focus to try to block CD16A shedding to maintain high levels of it on the cell surface to see what effects that has in cancer cell killing. So you can go about this in two ways. You can target ADAM17 directly, but one problem with that is ADAM17 is a pretty promiscuous protease. It has a lot of substrates, as illustrated in this slide, and this slide is actually fairly out of date. There's a lot more putative substrates that can be added to this list. And these substrates fall into several categories. There's cytokines, growth factors, receptors, adhesion molecules, and various other functions. So by targeting ADAM17, this can have various benefits, but it may have several adverse effects too, considering the number and various functions of these substrates. So a focus of ours has been trying to directly disrupt the cleavage of CD16A. So as I mentioned, ADAM17 cleaves CD16A proximal to the membrane. And on the left-hand part of the slide, I show the amino acid sequence of this membrane proximal region of CD16A where ADAM17 cuts. And through our work by isolating cleaved CD16A from peripheral blood natural killer cells and using mass spec analysis, we determined that ADAM17 cleaves in between this alanine and valine residues, as indicated by that blue arrow. And so it's known that the conformation of this juxtamembrane region is important for ADAM17 cleavage. So we tried to engineer a point mutation that might disrupt this conformation. So what we did is we exchanged that serine residue for a proline residue. And we found that this did effectively disrupt CD16A shedding. We refer to this construct as serine 197. That's the amino acid position of that serine 197 proline. But for this talk, I'll just refer to it as non-cleavable CD16A. So a couple of ways we went about looking at whether this receptor was non-cleavable. We used this cell line called NK92. It's a human NK cell line. But an advantage of the cell line is it lacks the expression of endogenous Fc receptors. But it's known that if you re-express CD16 in these cells, that they can mediate ADCC. So shown on the top left-hand side is a series of histograms where we stained parent NK92s for CD16. For other Fc gamma receptors, we show CD64. And these antibodies did not stain above an isotype control. So very little to no expression of these receptors. If we transduce in wild-type CD16 or our non-cleavable version of CD16 into these cells, now we find that CD16 stains very well. Our other antibodies to additional Fc gamma receptors don't stain above isotype control. And on the right-hand side with the red and blue histograms, what we did is we took NK92s expressing wild-type CD16A or the non-cleavable version. And for cells expressing wild-type histogram in the top plot, the red histogram indicates resting NK92 cells that express high levels of CD16. But when you activate these cells using PMA or cytokines or by ADCC, as represented by the blue histogram, we get a very appreciable downregulation in CD16A expression. But for NK92s expressing our non-cleavable version of CD16, again, the red histogram indicates expression levels on resting cells, and the blue histogram indicates expression on activated cells. And there's very little downregulation of CD16A. So by this assay and various other assays we performed, certainly indicated that our non-cleavable version of CD16A was not being downregulated. But since we engineered a mutation in the CD16A, we wanted to make sure we didn't, in some manner, decouple the receptor from its signal transduction pathway. So we looked at whether NK92s expressing our non-cleavable CD16 could mediate ADCC. And our ADCC assay is just depicted on the left-hand side, where we used a target cell, a tumor cell called SCOV or SCOV3. This is an ovarian cancer cell line. It expresses high levels of the tumor-associated antigen HER2. And so Herceptin is a therapeutic antibody that recognizes HER2. So we'll take the tumor cells, we'll add antibody, we'll add NK cells, and we'll look at the level of target cell killing. In our target cells, we've just labeled with an indicator that when the cells die, this indicator is released and allows us to quantify cell death. And so in the bar graph on the right, we're just looking at levels of cell death by our target cells. And the red bars indicate NK92 parent cells. They have no CD16. In the presence or absence of Herceptin, we get very little target cell killing. But for NK92s expressing our non-cleavable CD16 in the presence of Herceptin, very high levels of target cell killing we observe, and in the absence of Herceptin, much lower levels. So significantly higher levels of ADCC by NK92s expressing our non-cleavable CD16. So it indicates that our non-cleavable CD16 construct is functional and can transmit intracellular signals. So we wanted to move past this NK92 cell and see if we could express our CD16 construct in a primary NK cell and look at its function. So we can do this by two approaches. We can isolate NK cells from the peripheral blood, transduce them with a lentivirus to express our CD16 construct, or we can transduce a pluripotent stem cell with our construct and then differentiate these stem cells into hematopoietic cells and then eventually into natural killer cells. So all mature cells are derived from stem cells, and types of stem cells are distinguished by their potency or their differentiation potential. Totipotent cells have the greatest potential for differentiation. Embryonic stem cells are derived from the inner cell mass of the blastocyst, and they can differentiate into nearly all cells. And these have been used considerably for deriving primary cells to study their function, and also is now being incorporated in the clinical setting in clinical trials in attempts to replace damaged or diseased tissue. More recently, there's been an accelerating interest on induced pluripotent stem cells. And so pioneering studies by Yamanaka's group led to the discovery that somatic cells could be reprogrammed to a pluripotent state by overexpression of certain transcription factors that are indicated in this figure. And so then these induced pluripotent stem cells can be differentiated into hematopoietic cells and then into natural killer cells. So either embryonic cells or induced pluripotent stem cells have provided important platforms for studies of cell development, as well as the production, again, of primary cells that are being used in clinical trials as therapies to replace diseased and damaged cells, and also to generate novel immunotherapies. So Dan Kaufman at the University of Minnesota's group was the first to show that you could derive NK cells from human embryonic stem cells and induced pluripotent stem cells. And these cells possess a mature phenotype for the most part. They secrete cytokines. They are directly and indirectly cytotoxic against hematologic and solid malignancies in vitro and in vivo. And again, an advantage of this approach is these induced pluripotent stem cells are very amenable to genetic engineering, where you can incorporate genes or you can edit endogenous genes. And we've done both by expressing our non-cleavable version of CD16 or other versions of CD16 in these cells. And we've also used the CRISPR approach to edit endogenous genes. For example, we've knocked out ADAM17 in these cells. And then Kaufman's group had shown using various cytokines and feeder cells that you could differentiate these iPSCs into CD34 cells. And with further cytokines and perhaps feeder cells, you can differentiate these hematopoietic cells into early and mature natural killer cells. And then you have to be able to expand these natural killer cells to achieve numbers that you can use in your assays in vitro or your preclinical assays. And the hope eventually is to be able to expand them enough where they can be used in patients as well. So expanding these NK cells is, an important step. It's a difficult step, and a lot of different approaches have been used. And here I summarize 4 different approaches that have been used to expand NK cells. And this is nicely described in this review article by Granzin et al. In Frontiers Immunology. And so there's the use of cytokines, IL-2, IL-15, IL-21, or using them in various combinations. You can get some NK expansion. You certainly get NK cell activation. The expansion isn't all that great. You can use surface-attached antibodies. These antibodies recognize various activating receptors on the NK cell. They can be attached to a surface like a tissue culture plate or beads, and in combination with cytokines can result in NK cell expansion. And R&D systems, as Scott had pointed out, sells a product, this cell ex vivo human NK cell expansion kit, that uses an approach like this. We've used that kit. It's worked very well for us. Other approaches are taking autologous accessory or feeder cells plus cytokines to stimulate the NK cells. This results also in a very efficient expansion of NK cells. For example, you can just isolate peripheral blood mononuclear cells, and you can deplete the T cells and the B cells, and you can hit them with cytokines that favor NK cells like IL-15 and other cytokines and get expansion of the NK cells. Or you can take those peripheral blood mononuclear cells, deplete the B cells, and hit them with an antibody that stimulates T cells. T cells then release cytokines that activate the NK cells, and you get their expansion. A very well-utilized approach more recently has been using these allogeneic feeder cells, also referred to as artificial antigen-presenting cells, in combination with cytokines. So again, these pioneering studies by Carl June and Crystal McCall and Dean Lee had used these artificial APCs to expand T cells and expand NK cells. Again, a very efficient approach. And more recently, Dean Lee's group has developed these artificial APCs that are K562 cells expressing a membrane version of IL-21 and certain activating ligands like 41BB ligand, which is CD137 ligand. These cells then are irradiated, co-cultured with your NK cells, and results in a very efficient stimulation, activation of your cells. So we've really used 2 approaches. We've used the artificial APCs, and we used R&D's system as well. Both systems in our hands result in very efficient expansion of NK cells and high levels of CD16A expression. But it's important to remember that each approach for NK cell activation has some different effect on the NK cell function and the expression of their phenotypic markers. So if you're interested in NK cells, if you're interested in ADCC, some of these approaches can increase their natural cytotoxicity, effector function, and thus can mask more so the ADCC effect. And so you have to be, cognizant of this and careful which types of expansion approaches you use. This is just a snapshot of where we generated NK cells from induced pluripotent stem cells. We transduced them just with in a sham manner, just with a vector alone. And so, again, this is a busy slide, but we compared our derived natural killer cells to freshly isolated, enriched peripheral blood NK cells. And if you look at the CD56, CD3 staining, again, you can see that our derived NK cells are primarily CD56 positive, and they are CD3 negative, as are the enriched peripheral blood NK cells. There's a small subset of CD3 positive and a very small subset of CD56 positive, CD3 positive cells in there. And then we looked at various activation markers and inhibitory receptors, activation receptors and inhibitory receptors on these NK cells. And we find some of these receptors are expressed at similar levels to the peripheral blood NK cells, and some are differentially expressed. It was interesting when you look at the KIR receptors at the bottom that our iPSC-derived NK cells didn't express any of these KIR receptors, but they do express some of the inhibitory receptors such as NKG2A. So really, in a nutshell, our expanded NK cells have the phenotype of NK cells, of peripheral blood NKs. They're CD56 positive, they're CD3 negative, some differential expression of activating and inhibitory receptors. We think the lack of inhibitory receptors in our case is probably an advantage. It's just one less checkpoint inhibitor that these cells express. As indicated in this highlighted box, our cells do express CD16A. The majority of our derived NK cells express CD16A, where really essentially all mature NK cells in the blood express CD16. So we wanted to make sure our cells were functional and did mediate ADCC. To do this, we did this in vitro ADCC assay as depicted on the left, same as I described before. SCOV3 cells incubated with Herceptin. We throw in our iNK cells, our iPSC-derived NK cells, and look at the level of killing. In the gray bars on the graph to the right, we're showing ADCC by peripheral blood NK cells. If you add peripheral blood NK cells plus Herceptin to the SCOV target cells, we get nice efficient level of killing. If we throw in a CD16 blocking antibody to block the effects of that receptor, we see a significant decrease in ADCC. And if we do the assay with just NK cells and SCOV3 cells, but no Herceptin, we get very little killing. And the same thing is true for our iPSC-derived NK cells, as shown in the blue bars. In the presence of Herceptin, we get high levels of target cell killing. This can be blocked by an anti-CD16 function-blocking antibody, and very low levels of killing if we don't have the therapeutic or Herceptin antibody in there. So it indicates that our cells are cytotoxic and can mediate ADCC. So then we utilize iPSC cells by engineering them to express our non-cleavable version of CD16A. To do that, we just transduce them with a Sleeping Beauty transposon elements for a more non-random integration of our gene into these cells. And this vector also expresses GFP, and it has a selectable marker, zeosin. So we can select for the transduced iPSC cells. They will express green, so we know we're working with these cells. We can differentiate them to CD34 positive cells and then into NK cells. And so our NK cells express our non-cleavable version of CD16, and they also glow green due to the GFP. And so just in this figure, I'm just showing that. Again, if we compare these various phenotypic markers that we looked at before for iNKs expressing our non-cleavable CD16 versus iNKs that are just expressing the vector alone, essentially all the phenotypic markers are the same. But if we look at CD16A, we now find that cells expressing our non-cleavable version of CD16A express very high and homogeneous levels of CD16. And if we activate iNKs expressing endogenous CD16 or expressing our CD16 non-cleavable variant, we find again that the control NKs in this histogram plot, the red histogram shows high levels of CD16A. It's downregulated when you activate these cells in various manners. However, iNKs expressing our non-cleavable version of CD16A do not downregulate the receptor when they're activated. And it's interesting because we know iNKs express endogenous CD16A, but when we overexpress our non-cleavable version of CD16A, we find that we get a downregulation of endogenous CD16A and primarily just expression of our construct itself. And I think this is a fortunate coincidence. We're not exactly sure why that occurs. That was something we were looking into. So here we wanted to look at the function of our iPSC-derived NK cells in vivo. To do that, we used a mouse model. We can't use traditional mice because CD16A is really a different receptor in mice, and it's not a cleavable receptor in normal mice. So we have to use this xenograft model where we use an immunocompromised mouse and we implant into it a human tumor and then inject human NK cells, such as our iPSC-derived NK cells. Our tumor, in this case, as depicted on the left, is a Raji B-cell lymphoma. It expresses a tumor antigen CD20, and that's recognized by this therapeutic antibody, Rituxan. And so you implant this tumor. This tumor has been engineered to express firefly luciferase. It emits light, and you can see in the mouse images that over time we get more luminescence as the tumor grows. And so these mice that we're using, this is an immunocompromised mouse. It's a NOD-SCID gamma mouse. It lacks mouse B, T, and NK cells, and then we can infuse in our own human NK cells. If we treat these mice with tumor and Rituxan antibody by itself, the antibody by itself does have some effect on the tumor. It diminishes its size, but again, you can see over time the tumor does continue to still increase in size. If we administer in these mice just the NK cells expressing non-cleavable CD16, this doesn't have much of an effect on the tumor. You can see the tumor continues to grow over time. However, if we inject into these mice our iNK cells plus rituximab, our Rituxan antibody, you see a very appreciable diminishment in tumor cell growth, indicating that our iNK cells expressing non-cleavable CD16 are functional, and in this in vivo ADCC setting are able to kill tumor cells. So I was running short on time, so I was really just going to leave it at that. I really just wanted to present some of the work that we're doing on NK cells and some of the approaches that we're taking to try and enhance their function for generating, with the goal of generating a new, hopefully improved type of cell therapy for cancer. And again, some of the approaches we're using is expressing our constructs in primary NK cells, deriving these NK cells from induced pluripotent stem cells, and then trying to proliferate these cells so we have cells to use in our in vitro and in vivo assays. So as a brief conclusion, I just wanted to remind you NK cells are cytotoxic lymphocytes. They use an extensive array of germline encoded receptors that induce activation and inhibitory signals. CD16A is one of their most potent activating receptors and has an exclusive role in mediating antibody-dependent cell-mediated cytotoxicity. Cell surface levels of CD16A are regulated by ADAM17. So ADAM17 appears to play a checkpoint inhibitory role in its regulation. Preventing CD16 shedding is intended to maintain its surface expression and function during ex vivo expansion of these cells or when these cells are in an immunosuppressive tumor environment. And iNK cells provide an opportunity to assess the effects of preventing CD16 shedding on various NK cell effector functions in vitro and in vivo. And take-home message is, we feel that these iPSC-derived NK cells provide future opportunities for a cellular immunotherapy. They have various advantages. iPSCs are highly stable, genetically modifiable clonal cell lines. There are defined methods for producing NK cells and expressing enhanced receptors in them. And these iPSC-derived NK cells, they can be autologous, they can be allogeneic, they're scalable for an off-the-shelf therapy, and they circumvent various issues of isolating NK cells from individuals, such as the process of isolating the NK cells itself, donor selection. NK cells are a very heterogeneous population, and there's the risks of also isolating and expanding contaminating T cells and B cells. And so our non-cleavable CD16A is the basis of a patent application and has been licensed by Fate Therapeutics as a clinical stage biopharmaceutical, which is a clinical stage biopharmaceutical company, and is focused on the development of programmed cellular immunotherapies for cancer and utilizing the non-cleavable CD16 as a platform for generating primary NK cells from iPSC cells as a potential cellular therapy. So I just wanted to conclude on this acknowledgement slide, acknowledging individuals in the lab that played a key role in generating some of the data that I show. Some of our important collaborators here at the university, Dan Kaufman, who actually now is at UC San Diego, Jeff Miller, who oversees a large NK cell biology group and clinical program here at the university. Jimmy Wu has been a key collaborator with his expertise in FC receptors in generating engineered iPSC NK cells. I want to thank or recognize our centers and institutes that provide a very collaborative environment so these studies can be done. Our Amazonic Cancer Center, Center for Immunology, Stem Cell Institute, and of course our funding sources, which are essential for our discovery research and generating new therapies. And so I'll leave it at that and see if there's any questions.
Okay, great. Thank you very much, Dr. Walcheck. What a great seminar, very informative. Some fabulous information there and good insights into what you guys are doing in your lab. And we'll move into the Q&A session here quickly. I just want to remind the listeners that if they have questions for Dr. Walcheck, they can enter them in the Submit the Questions box or Ask a Question box that's just below the presentation screen. And I'll relay these to Dr. Walcheck as they come in, and we'll try to address as many as we can in maybe 10 minutes or so. So let's just get going. So I think the first question that came in is related to a point that in the summary that you had. They were asking about what the advantages of using iPS-derived NK cells would be over using peripheral blood NK cells. And I think you addressed that a little bit in that they provide like a sustainable source and off-the-shelf option that circumvents some of the donor issues that you would get with peripheral blood NK cells. But if there are any other issues around advantages of using iPS iPSCs, drive NK cells over peripheral blood that you could think of. And in particular, they're interested in the efficacy. I think they're referencing the slide looking at the specificity releasing assay, the ADCC assay, where you compared PB NK cells versus iPSC NK cells, and the iPSC NK cells were lower. So if you can comment on that too.
Sure, yeah. I think that for us, being a basic research lab, I think the biggest advantage of the iPSCs is, again, their ability to genetically modify these cells, either alter endogenous genes or express new genes in them. They're very amenable to that and still maintain their pluripotent state, and then can be differentiated, in the CD34s and NKs or various other cell types. So that's a big advantage for us, and the techniques to be able to differentiate these cells are well described in the literature, very reproducible. They've worked well for us and our work with Jimmy Woo in terms of doing that. So that's been a big advantage. In terms of efficacy, or I guess compared to a peripheral blood NK cell, as I went through it pretty quickly, but the FACS plots I showed, I mean, they don't express at the same percentage of positive cells CD16A compared to peripheral blood NK cells. The majority of our iNK cells are CD16 positive. We're essentially all mature peripheral blood NK cells are CD16 positive. So I think because of that, in our in vitro ADCC assay, we see a little less ADCC. However, when we express, and this is work done by Fate Therapeutics as well, when you express the non-cleavable version of CD16A in these cells, they actually mediate higher levels of ADCC in vitro and in the animal models. So they seem to work better than the peripheral blood NK cells.
Great. Okay. Another question, more generally related to cell therapies. Are there companies or universities that are already using NK immunotherapies clinically? Yeah,
that's certainly been done at our university in various ways. Pretty much patient or donor-derived NK cells, or trying to stimulate a patient's own NK cells. So adoptive transfer or stimulating a patient's own NK cells, in particular after bone marrow transplants. Other universities are doing this as well. I know MD Anderson, various institutions are working on these adoptive cell therapies, as well as trying to stimulate NK cells in the patient. Work being done at the NIH as well.
Okay. This question asks if you've identified the phenotype of ADAM17 or of ADAM17 knockout in your iPS-derived NK cells.
Yeah, we were interested in that to see if we could do it, and also we were a little concerned that might mess up the stem cell or the derived hematopoietic cells or NK cells, because in that slide I showed of an overview of the various ADAM17 substrates. It has a very broad role in cell function and cell development. And so we did knock out ADAM17 using a CRISPR approach, and we found that didn't seem to have an adverse effect on the iPSCs. We were able to derive hematopoietic stem cells and NK cells, and so we didn't seem to disrupt that process, at least at the level we've looked at it. We could get NK cells, And we just haven't spent a lot of time on those cells because we've been more focused on expressing our CD16 constructs in NK cells. So, yeah, I don't have a lot of information on that. We know we can knock it out, and we know we can derive an NK cell, and we know they don't shed their endogenous CD16. But we haven't looked at it too extensively.
Sure. Okay. And related to that would be, I guess, a similar inquiry about doing the same thing in NK92 cells. So if you've assessed a knockout or knockdown of ADAM17 in those cells, and if there's effect on viability and cellular toxicity.
Yeah, most of our stuff, we start with an NK92 because, again, it's a fairly easy cell to work with. They don't have FC receptors. But for ADAM17, we had not knocked it out in an NK92, so we haven't looked at that. We have a lot of ADAM17 inhibitors, so we tend to just use those instead. But we were curious about that, knocking it out in NK92s and then expressing various ADAM17 substrates in those cells in addition to CD16. But at this point, that's still on the back burner for us.
Okay, great. The question here is, how does the absence of KIR expression affect your inducible NK cell function?
Yeah, that's interesting. And the expression of KIR really varies on how you derive these NK cells. I know earlier studies by Dan Kaufman, again, who initiated this work here at the university, they would get KIR receptor expression of some of the KIR receptors. I believe Fate Therapeutics sees KIR receptors as well. By our particular approach, we've modified this differentiation process in various ways. And so we're not seeing, at least the few KIRs that we've looked at. We haven't done, in terms of functional assay, much more than just the ADCC assay, because that's what we're interested in. So we haven't looked at their natural cytotoxicity function that much. But in that slide where I was showing in vitro ADCC plus and minus the Herceptin antibody, in the absence of that antibody, we were getting very low levels of killing. SCOV cells really aren't the best target for natural cytotoxicity either. They don't have a lot of ligands for these KIR receptors. We've looked at other tumor cell lines, other ovarian and B-cell lymphoma cell lines, and we don't see a lot of natural cytotoxicity, and I think that's in part due to the lack of these KIR receptors. So I think for us, again, I think that's an advantage, not having them and not having an additional inhibitory process. So we're maximizing the CD16A signaling and function. But again, it's something we haven't looked at a whole lot. I think other labs here at the university are probably looking at that more closely. But yeah, I don't really have a lot of information that I can share on that.
Okay, great. We're getting quite a few questions in here, but I think in the interest of time, maybe we'll handle maybe 2 or 3 more, and then maybe offline I can work with Bruce to get some of the answers out for some of the ones we can't address here live today out to those people who submitted those. So let's go through just maybe 2 or 3 more. The first one here asks, as well as modifying CD16 so that it remains on the NK cell surface post-activation, could you also engineer the NK cells to express the high-affinity 150AV variant at the same time, thus providing 2 mechanisms for increased efficacy?
Yeah, that's a good point. I'm glad they brought that up because I kind of glossed over that. I forgot to mention that our non-cleavable version of CD16, we did engineer the valine residue in them. So it is the higher affinity version of CD16, plus it's non-cleavable. So it has both of those benefits. So our receptor is the higher affinity version. But for the endogenous CD16A, you could certainly do that, again, through a gene editing approach. You can modify the endogenous gene. It's a little tricky because these cells also have the CD16B gene, which is very similar to the CD16A gene. So you're, in effect, going to be modifying both of them. And then you have to do that on both alleles. But there is a group here at the university that is using a CRISPR approach to modify the endogenous gene, initially to engineer the mutation that we described to make it non-cleavable. That is one problem with, I guess, if you were to do this type of engineering with peripheral blood isolated NK cells, because the majority of individuals are not homozygous for the V variant. They're either heterozygous or homozygous for the F variant. So they're not going to have, in most cases, that higher affinity version. So either you would have to edit the gene for that as well, or again, using the induced pluripotent stem cells, where at the same time we can just insert this gene into those cells.
Excellent. Okay. Next question is, are there specific ex vivo expansion protocols better suited for induced NK cells with and without non-cleavable CD16A? And have you correlated which protocols upregulate ADAM17 and correlate that with effector function?
Yeah, so for ex vivo expansion, again, that's, I think, probably the trickiest part of this, because there are many ways of doing that, and they all have different effects on the NK cells. And in the end, you kind of generate an NK cell that kind of has an immature phenotype, where maybe you do get loss of these key receptors, and you get upregulation of the CD56 marker. But again, they're still cytotoxic functional cells. To use the artificial APCs is a nice approach, but either you're going to have to generate those cells or acquire those cells, which can be tricky, but there's a lot of good literature out there on describing these cells. And I think, again, in my mind, I think the best commercially available approach, again, is using R&D's expansion kit, which works very well and maintains CD16 expression on these cells. Because they're getting hit with cytokines, I think you do have higher levels of natural cytotoxicity as well. But that's been a good approach for us. And so what the question was, I think with or without CD16, I think those artificial APCs or R&D's kit works just fine if the cells do or do not express CD16. We haven't so much looked at ADAM17 upregulation or change in expression following expansion. We know it's there, it's expressed, and it works. But we haven't looked at so much its expression levels. And it's really regulated in two ways. It's an inducible protease, so following cell activation, it just gets turned on. I didn't really get into the mechanism of that. And it can be upregulated, and that likely affects its function as well. And we've spent more of our time on this induction mechanism, what turns it on, but have not looked at that too closely in NK cells, more so in neutrophils.
Great. Okay, and we'll just do last one here for the day is more on the cell therapy angle here. And they're asking, what levels of expansion do you see as necessary to reach therapeutic levels for human treatments?
Yeah, I'm not the best person to answer that. I know with the Fate's iPSC to NK cell approach, they can really get a million-fold expansion. They can convert one iPSC into 1 million NK cells. And I think they primarily use the artificial APC approach. I think to dose a human, they're typically using 10 to 20 million NK cells per kilogram. And so you definitely need good expansion. And I think based on the literature, the artificial APC approach using K562s are these EBV-derived cells are capable of expanding the NK cells enough so they can be used in the clinic. It's certainly being done in clinical trials, developing these expansion approaches, and they're certainly, I guess, deriving high levels of these cells that can be delivered to patients. But I think it really depends on the type of cancer. In our case, we're trying to enhance the efficacy of a therapeutic antibody. Many types of therapies are just administering natural killer cells themselves to utilize their natural cytotoxicity. We're trying to take advantage of their ADCC potential and then trying to, again, hopefully down the road, increase the efficacy of these therapeutic antibodies. That may require less NK cells because we also have the benefit of the therapeutic antibody. But there are some very good reviews out there, and in my talk I mentioned that Granzin review. That covers this topic very well.
Great. Okay. Thanks a lot, Bruce. So I think in the interest of time, we'll leave it at that for the Q&A. There are a couple here, and I'll work with Bruce to maybe address those offline and get answers back to you guys via email. But this webinar should be available on demand and sent out to all the listeners tomorrow, so you can review and listen to it again. And thank you all for attending today. I appreciate it. Thank you, Dr. Walcheck, for contributing. And have a great day, everybody.